I'm going to demonstrate the Colony PCR lab. In this lab, I'll use a process called PCR, or polymerase chain reaction, to determine if RFP is present in the white and pink colonies from our transformation lab. In part A, I'll perform PCR to make lots of copies of the RFP gene if it's present. This is called amplification. In part B, I'll use gel electrophoresis to analyze the PCR products and determine if the RFP gene is present. Here are the reagents and equipment that I will use to conduct part A of the lab. I have a tube of PCR master mix. This contains a combination of salt solutions and buffers that are necessary for the PCR reaction. I have a negative and a positive plasmid control. The positive has the RFP gene, the gene of interest. The negative does not. And then I'll use pink and white colonies from the plate from our transformation lab. And I'll set up 4 reactions in these PCR tubes. One with the positive control, one with the negative control, one with a white colony, and one with a pink colony. Once these reactions are set up, I will place them in the PCR machine, which is called a thermocycler. It will go through many cycles of heating and cooling the samples, and this will help to make copies of the DNA or amplify the DNA. First, I need to label the tubes 1 through 4 and with my initials. Now I'm going to set up my reactions. Everything needs to be kept on ice. So when I use the tubes, I'm only going to touch the very top of them. And you can see in here we have ice to keep the tubes chilled. Each tube will get 23 microliters of the PCR master mix. Next, I'll add a red colony to tube 1, a white colony to tube 2, 2 microliters of positive plasmid to tube 3, and 2 microliters of negative control plasmid to tube 4. I'm using gloves to help prevent contamination. I'm going to use a clean tip to gently scrape a red colony off the plate and add it to tube 1. Then I'll use a clean tip to add a white colony from the plate to tube 2. Once everything is added to the tubes, I'll look closely and check for big bubbles. If there are bubbles, I can centrifuge the tubes. If not, they're ready for the PCR machine. Now I can take my samples to the thermocycler to run. The screen shows the changes in temperature as the thermocycler is going through many cycles. First, it goes through an initial denaturation at 95 degrees Celsius for 270 seconds. Then it goes through 30 cycles consisting of denaturation at 95 C for 30 seconds, annealing at 53 C for 30 seconds, and extension at 68 C. After the 30 cycles, it goes through a final extension at 68 C for 300 seconds. Here are the equipment and reagents that I will be using for part B. I've got my gel electrophoresis already set up. It'll be a very similar process to when I've used gel electrophoresis in the past. I have my DNA ladder, and then I have my 2 control samples and my 2 experimental samples. Because the master mix has dye in it already, we don't need to add additional loading dye. Just like in past gel electrophoresis labs, I'll be loading 10 microliters of each sample and the DNA ladder into separate wells in the gel. Now I'll press on and run the gel at 100 volts. Similar to other labs, I need to watch and look for when the yellow dye is halfway to 3/4 down my gel. That's when I know to turn off the gel and look at my results. There's gel green in the gel, so I'll be able to visualize my DNA on a blue light source or using a UV light. Using these results, we can determine if the RFP gene is present in the red and white colonies from our transformation lab. Again, I'm not going to give away the results of the lab. You're going to have to check with your teacher to see a gel from your class.